I would like first to thank all the organizer for the invitation. So actually, yeah, it will be a bit redundant with what humans say, but it will be the same talk from a mathematical point of view on a really specific detail of what you speak about. So, let's get some basic information about the prion that us as at least me as mathematician, I understood. So prions are like a transmissible spongiform encephalopathy. So they have three characteristics. First is like the progression. So as Human says, the incubation periods is really long for prion, and it has also a lot of variability. So for instance, for like the VCGD, the incubation period can take 1 to 30 years. Okay. Then also like the prion diseases are transmissible within a species, but also between species. And the most famous example of this kind of transmission is like the bovine spongiform encephalopathy, which is also called the mad cow. So it came from bovine, and then it got it got to humans. Okay, and also yeah, these diseases are fatal. So what do they do actually? So in this picture, it's taken from a study of Koizumi. Okay, you have like some kind of rare IRM, MRI, sorry, images of a woman of 64-year-old who was infected by the prion disease, the VCGD. So in the row A, it was 5 years before the symptoms. In the row B, it was 6 months before the symptoms. In the row C, it was 2 months before the symptoms. In the row D, it was 3 months after the symptoms. Okay, and what do we want to look at? It's the white spots. The white spots are the hyperintensity of the signals, and which also indicates neuronal loss or lesions. And what we can see is that the woman has already like this disease like 5 years before she has the symptoms, and then there is not much change like during the 5 years, but right after she got the symptoms, then there is a huge neuronal loss. So it also like illustrates the incubation period, but also the non-linearity of the phenomena. So it's not linear, like you will not have like a linear degenerations, but you have like will have like some kind of acceleration. Okay, and the question is, how can we understand this process? How can we stop the neuronal loss and so on? But this is still an open question. So in order to solve these questions, we look at the phenomena. So as Human says, the prion disease are like caused by a complex phenomenon, which is like a protein polymerization. So there are two kinds of proteins in the term of prion. You have the PRPC, which is an LC1, and it does not cause effect to the organism. And then you have the PRPSC, which is the pathologic form of the protein. And then when the PRPSC aggregate and start to form like fibrils, then you have like a neurodegenerative disease and so on. And what you can see on the atomic force microscopic image is that you have one fibril, and the red dots are like the monomeric elements. And you can see the composition of the fibril is then an elongation or like a shortening of this kind of fibril by its end. Okay. So now the atomic force images are like static data. So we understand like how we can model this kind of polymerization process, but what we really want to understand is how does it evolve through times. So it's about the kinetics, and then I will speak about like the experiments that Human showed you. So this is a static light scattering experiment. So what they do is like they put prion aggregates into a test tube, and then they let it flow. And what they do, they have a light, and they record the reflection of the light under a certain angle. So how can we assimilate this signal with the size distribution? actually we can say that this signal is exactly like the second order momentum of the size distribution. That's what's written here in the discrete size setting, here in the continuous size setting. So when you have like a big decrease of the signal, that means that your size distribution is localized around big polymers and it goes to smaller clusters. Okay. And what's interesting in the humans experiments is that when you zoom, like for example, you take a look at C, it's a zoom between 4.1 hour and 4.3 hours, then you can see like harmonic oscillation in this signal. And when you do the same zoom, but much later, so for example, in E, between 15.3 and 15.5 hours, then what you record is only noise, okay? So what does it mean for this kind of signal to have oscillation? That's really tricky because it means that you have some kind of transport toward the small clusters, big clusters, and again, small clusters, big clusters. So it's really, it's really intriguing from a mathematical point of view. And actually, that was the start of the collaboration. How can we explain with a size structure model this kind of oscillation? So we start from the simplest size structure model that we have, is the seminal model of Becker and Döring. It was introduced for like other kinds of aggregates. And what does it show? It says that you have like a monomer C1. When it encounters a monomer Ci, then it becomes a polymer of size Ci plus 1 with a rate, polymerization rate Ai. And then you also have the inverse, meaning that Ci becomes a polymer of size Ci minus 1 plus C1 at a rate Bi. Okay, so you have on the right a scheme about what it does. So you see like this, it's a network of reaction, chemical reaction, and it's always in both ways. Okay, so it has a kind of structure which is called detailed balance, since it's totally reversible. Okay. And when we write the mathematical equation of this system, then for the Ci, we have the difference between two rates. The first rate, Gi minus 1 minus Gi, where Gi is the net rate at which a cluster of size I becomes a cluster of size I plus 1. Okay? And then C1, we have a more complicated equation because C1 actually is intervening in all the reaction, either as a product or as a result for the depolarization. Okay, another important property of these systems, it's that there is a conserved quantity, which is the total mass. And actually, that's what we want, because in the human experiments, we are in a closed environment, so we do not lose mass. Okay. So how can we understand this system, understand its behavior, but then we look at asymptotic behavior. So in order to detail the behavior, I will only speak about the subcritical case, and I will explain what is a subcritical case. So what we do is, like, first we try to write the steady state distribution, size distribution of this system. So we introduce the detailed balance coefficients. It's just a way of rewriting the coefficient in order to have a much more simpler formulation for the steady state. In terms of QA plus 1 is the product of the ratio between the polymerization and the depolymerization coefficients. And then we have the steady states, which can be written as a geometric sequence. Okay. And this sequence should be summable. And that's what we call actually subcritical. In fact, this sequence, the sum IQI Z power I is linked. It's equal to the conserved quantity of the total mass. Then the subcritical case means that if initially you have a conserved quantity, a total mass which is inferior to the rate of convergence of this sequence, then you're subcritical. And I will, in this talk, I will only speak about the subcritical case. There are still open questions for the supercritical and so on. Okay, so now that we have the steady states, there was lots of work actually in the 80s about like the convergence about this system toward the steady state. And the first convergence theorem was proved by Bolkar and Penrose in 1986, where they introduced a Lyapunov functional. And then they proved that the system converged to this steady state. And then afterward, there was like a better result with a much more finer convergence rates by modifying like the Lyapunov functional. Okay, but in all these works, there were no oscillation. So this system has some kind of rigid structure since it's detail balance. It's totally reversible. Then you can always extract like a Lyapunov, and then you prove like the exponential convergence toward the steady state. So you cannot have oscillation. Then the question is, what are the simplest reaction networks which oscillate? Sorry, can you hear me? Okay. Also, if you want to interact, feel free to interact. It does not.
Maybe I have a very quick question. I was waiting to the end, but since you mentioned it, maybe.
Yeah, sure.
But I noticed those are names known in mathematics. So John Bohr, Pierre-Emmanuel Germain, there are no names. I don't know exactly what they do, but I know those names. And but you start with a very biological experiment and stuff. So my question, does the free energy are just Lyapunov functions to prove the results, or how they link to the real? Because free energy is also a real biologic stuff. So is there a strong link? Is there? But you're not if you don't want to.
No. Actually, it's kind of tricky. So I don't know. The question. Okay, they call it free energy. Maybe there is a link, but it was in a much more theoretical background, and meaning that the energy of the system is decreasing and get to a state where you have no energy anymore. Yeah. So that's my understanding about the free energy.
Thank you. We'll have a look at the papers. Or thank you, Mathieu. All right.
You're welcome. Okay, so I go back to the topic. Then, what are the simplest model that oscillates? So one of the model, which was actually introduced by one of my coworker, Juan Velasquez, it's a Becker-Döring with an atomization reaction, meaning that you have some kind of critical size, say big M, where when the cluster arrive at this size, then it disintegrates into M monomers. And then you can understand how the system oscillates, because you will reach this size, then you will get a source term toward the small cluster, and again. And they gave like a complete proof about this phenomenon in the paper in nonlinearity. But this kind of phenomenon is based on the atomization reaction, which is not possible in our case, because the prions are known to be really strong, structurally speaking. So you do not have like, there was also like some controversy about fragmentation. So you cannot have atomization reaction in this kind of stuff, at least not in a normal environment. Okay, so then we went back to the biology. So we were inspired by a simplified Belousov-Zhabotinsky model. And so we take like three components: V, which is like the non-pathological monomers, W, which is the pathological monomers, and C, which is the fibril. And then we have some kind of a competition between the three species. V plus W gives you 2W. That means that the pathological protein is infectious. It can infect like the non-pathological protein. It recruits it. Then you have W plus C give you 2C. That is, in a sense, the polymerization process. And then what we have is a C plus V gives you 2V. And that's actually the depolymerization process, but in a non-catalytic circular way. So when we put into this system all the size index, then we get this chemical reaction network, where V plus W gives you 2W with the rate K. Then you have W plus CI, which gives you CI plus 1 with the rate AI. And then CI plus V, which gives you CI minus 1 plus 2V with the rate BI. Okay. And it gives rise to this kind of system where you have like, now you have like an infinite size dynamical system, but there is some differences between the Becker-Döring one. The first one is we have like two kinds of monomeric elements, V and W. Okay. The second difference, which is is that the depolarization in this system is not as the same as the Becker-Döring one, because now it's catalytic. You need like to encounter like a species V in order to depolarize. Okay, so it introduces a new nonlinearity in the systems, and it can be seen into these terms. Now you have like a product term between V and Ci here. And the third difference is that C1 in this system does not play the same role as C1 in the Becker-Döring one. C1 here is the smallest size, is a concentration of the smallest size cluster. So C1 cannot depolarize, actually, and this gives rise to two conserved quantity. We still have m tot, which is the total mass of the system, which is conserved, but we also have the total concentration of the polymer, which is conserved also. Okay, and now in order to study this system, actually, it's quite hard to study like an infinite size ODE systems. So it's better like to have like a continuous settings. So what we do is like, okay, to consider that the size is continuous, then the difference between the two rates is actually a first order differential operator, okay? And then it gives rise to this system. So you have a transport equation, okay, linked to two ODEs, and actually it's a nonlinear PDE, the transport equation, because it depends on W and V, and the V and W here depends on the non-local term of the solution, which is the integral of BI-CTI or the integral of AI-CTI, okay? So I will just add some stuff here in this system. This system is okay, if we take the subsystem for the monomers, so V and W, actually it's closely linked to a Lotka-Volterra system where V plays the role of the prey and W plays the role of the predator. You have like these two terms, so minus KVW or plus KVW, which plays the role of the interaction. And then you have a source term for V and a depletion term for W, okay? And so the strategy in order to study this nonlinear system with the transport equation is to find some way to decouple the ODE system from the PDE. Actually, if we take some assumption on the coefficient rate, we can use the conserved quantity in order to have some decoupling. So that's what we do. So let's just consider that we have a linear reaction rate. Then we have a decoupled system for V and W, and actually the system for V and W is just a generalized Lotka-Volterra system. Okay. And then in studying these systems, we can prove that the V and W are periodic of the same period. And then the solution, it's just a transport equation with a speed which is periodic. And then the solution is also periodic and has the same period, okay? But this supposition on this hypothesis on the linear coefficient for polymerization and for depolymerization is not really realistic. What is more realistic is that the depolymerization is dominating in this system. So that's the next assumption, is like to take like a constant rate for polymerization, which is equal to 1 for simplicity, and a linear rate for depolymerization. And then we have some assumption about the total mass, meaning that the total mass, actually the mass on the polymer must be strong, like at least like the mass should not be totally into the monomers. Okay, that's like the inequality V0 plus W0 is inferior to m tot. Okay, we still have like the total concentration that we denote rho 0. And then the third hypothesis is about like some kind of, it gives you some information about the initial size distribution. So if you remember, if you neglect the mass in V0 and W0, then m tot is just the first order momentum of the size distribution. Rho 0 is the zero order momentum, and then m tot over rho 0 is an indicator of on which size the mass is most localized. And then that says so you can interpret the third condition as the one that the size distribution initially must be localized in the large cluster, at least bigger than 1 over k, where k, I recall, is just like the rate at which v becomes w. Okay, so if these three hypotheses are fulfilled, then we have an asymptotic convergence to our steady state in 2 steps. The first step is just to say like that the characteristic, actually the mass concentrate along the characteristics. And the second item of this theorem is to say that, okay, the solution converges to a Dirac mass where the size, actually the size is depending on the equilibrium of the monomeric ODE system. So how do we prove that? So first, we look at the ODE systems. So we have this kind of ODE, which is also a modified Lotka-Volterra. For this kind of ODE, we have a Lyapunov functional. However, the Lyapunov functional, the derivative of the Lyapunov functional is equal to 0 along a line in the phase space. Okay, so we need a bit more work to have a speed of convergence toward the steady state for this system, and we look at local estimate near the degeneracy line, and saying that the trajectory will cross this line but not get stuck to this line when it's far away from the steady state. Okay, and so if we have that, we have like an exponential convergence, but with a rate alpha that we cannot explicitate. Okay, and then the second step is to look at the characteristic and to prove that the characteristic converge toward one state. And so the first equation, which is like the first item of the theorem, state that you have like a convergence of you have the mass which is concentrating along the characteristic, and actually that's due to a result of Philippe Michel. And then the second item is just to use like the theory of asymptotically autonomous differential equation. Then V converge to V infinity, W converge to W infinity, and then these systems converge to W infinity over V infinity with exponential rate also. And finally, we can prove the convergence of the solution toward the Dirac mass with respect to the Wasserstein distance using these two previous results. Okay, so now for the continuous system, depending on the assumption we made on the coefficient, we either have like a sustained oscillation or like an exponential convergence towards the Dirac mass. Actually, that's not really realistic from a biological point of view. And actually, one of the mistakes we made is that the continuous approximation actually is a first order approximation. So we neglect all the diffusion term. So now we go back to the discrete size model. What can we say about the discrete size model? So the first is like for general reaction coefficient, we can categorize the steady states. So we have boundary steady states. The first one is where we have no mass into the monomer and all the mass is in the polymers. And this one is always unstable. Then the second steady state is like where the mass is in V. All the mass go in V and in C1, and you have no mass in W and the Ci bigger than 1. And this one is linearly locally stable only if actually the initial mass is localized toward the small clusters initially. Sorry. And then, if this condition is not fulfilled, if you have like initially some mass in the large cluster, then you have an existence of a strictly positive steady state. However, we do not have the convergence toward the steady state. Okay. So we need to make some assumption in order to have much finer results. The one assumption we made is that, okay, let's take all the reaction coefficients equal to 1, constant. And then what we do is that to say, okay, the total mass is equal to 1, but the total concentration of polymers is equal to epsilon. And so this has two impacts. So either like you have like initially lots of mass in the monomeric elements and a bit of mass in the polymers. And also the second consequence is that if you look at the size distribution of your solution, the size distribution is really localized around one size, which is big, which is around 1 over epsilon. Okay. And so we introduce this epsilon in order to do some perturbation analysis on the system. Okay. Then we can rewrite the system as the following. We still have the two equations for V and W. And then we use like actually the conserved quantity in order to simplify the equation. And what we have for V and W is like we have a Lotka-Volterra systems, but you have a non-autonomous perturbation only in one equation, which is C1. Okay. And again, we can rewrite like the equation for GI. And then if we we can rewrite in such a way that we have a discretized transport term with a rate W minus V, and you also have a discretized diffusion term with a rate W plus V. And once we have this system, we introduce some characteristic quantity. We first take a look at the energy, which is actually the first integral of the non-perturbed Lotka-Volterra system. And then when we look at this energy, actually, it's not monotonous. It will change sign, okay? But what we want to look at, okay, I will speak about this later. Then also we can look at the period T, and T can be rigorously defined with a Poincaré mapping, meaning that if you look at the phase space V and W, you can write a half line starting from V infinity, W infinity, towards plus infinity, and then you look at the time it takes to the trajectory to cross this line, and this will give you the period. Okay. On the complete system, we can actually write the steady state. Then we have C1, which is of order epsilon square, Ci, which is still a power series which depend on C1, V, which is of order epsilon, and W, which is of order epsilon minus epsilon square. And then we introduce two important quantity, which is like the cluster length, which is the size in space at which the mass is most localized. And then we assimilate the cluster distribution to a function phi. We say that Ci is equal to epsilon over L of phi I over L, where phi is a function which is integrable with integral which is equal to 1. Okay, and then you see that L is also some kind of dilatation coefficient for phi. So if you take phi as a Gaussian, then L will be like the spreading of the Gaussian. Now, the goal in order to really understand our system is to understand the evolution of the energy, the period, the characteristic length L, and the change in the shape of phi at each iteration of the Lotka-Volterra cycle. And we can have a complete description of the behavior in 4 phases for this system. So this is a summary of the results. So if we take a look, the blue graph is just like a representation of the evolution of the energy, and the black graph is a representation of the evolution of the characteristic length. So in the first 1 over epsilon cycles, the energy is equal to 1. So you have no change in the systems, but actually the characteristic length is growing towards 1 over epsilon. Okay, then in phase 2, once your size distribution is sufficiently spread, then the impact of C1 will play a role and it will decrease the energy. Then in phase 3, the energy is decreasing like a bit faster. And in phase 4, the energy is constant equal to epsilon cube power 3. And the cluster length still is 1 over epsilon, but you have like a convergence toward the steady state. So I will explain the phase 1. So the phase 1 is the epsilon, the energy is equal to 1, and the period is around like for one cycle, 1 over epsilon square. And what we do actually is to take a look at the phase space of V and W, and we try to understand. We separate the phase space in some sub interval, and we understand the changes in the size distribution. But for instance, actually, the graphs are to be taken by two. So the left one are the phase space VW, and the right one are the evolution of the size distribution. So what you see is like when W decreases and V also decreases, but is really small, then you have the size distribution will spread with diffuse and go to the large cluster, okay? And then you have like some kind of a long phase where V and W are really small, but V is growing, and then nothing happens. And that is because the transport term and the diffusion term depends on V and W, okay? And then again, you go back to the cycle. So you have like the transport term, which is dominating. And then you have your size distribution, we go back to the small size cluster. And then the most tricky part is on this line, where it goes really fast. And then you have like first it goes to the smaller cluster, then it goes back to the larger cluster. So in this phenomenon, you have like different time scales, as I said. Like you stay most of the time in the phase where nothing happens, no transport and almost no diffusion, okay? And that's why actually, in terms of asymptotic analysis, that's why actually the energy stays constant. But at each cycle, you will have some kind of spreading. And so at each cycle, actually, the size distribution will spread. Okay. And so in order to analyze this phenomenon, we can rewrite as a continuous equation the phenomenon. And then we have this kind of transport advection diffusion equation. Okay. And as I said, we divide the phase space for V and W into several time interval, and we have like two interval from 0 to delta t and from t0 minus delta t to t0, where it's a really fast, it's a really small intervals, and you can neglect like the diffusion process, and you only have the transport processes. And actually, most of the time, you have a pure diffusion process, okay? And then the next step is like to say, okay, how does the size distribution evolve? So then you look at the phi, and what you can say is like using this kind of terms, this kind of solution into the equation. Okay, sorry. Okay, using this kind of time interval, then we can write the solution as the following. So the phi1, so the next iteration of the size distribution will be like a convolution between a heat kernel and the solution before, okay, plus a Dirac mass multiplied by one term. And actually, I will go back, I will explain what this term is. Okay, when the size distribution reaches C1, or like the size, the really small size, since we have a concentration of we have a conservation of the total concentration of polymers, then we accumulate the mass into the first polymer, sorry. And then we have like two kind of profile. We have a direct mass and then the Gaussian, which is due to the diffusion process. And then again, when we have the transport kicks in and the transport is going toward the large cluster, we also have a diffusion. And then we have the diffusion of the direct mass and still the diffusion of the rest of the size distribution. And we have this kind of profile for the size distribution, okay? And now this is an example, a numerical example about what happens. So these are like 4 iterations of the Lotka-Volterra cycle. So you see at 0, so when it's black, it's at time 0, okay? And what you can see is actually, it's like at the beginning, you have a Gaussian profile, okay? And then afterwards, you have a diffusion, but it's not symmetric. And this is because of the concentration on the Dirac when you reach like the small clusters, okay? And so now you can see how it will converge to an exponential decaying size distribution. Okay, and then in phase 2, when the concentration is when the spreading is sufficiently big, then that means the interaction with C1 it's much more frequent, and then the energy will decrease. And we have actually we can quantify how this energy decrease. And again, actually, this term is actually half of the mass, which concentrates on the negative half part, which is then, so you have like a diffusion process toward the negative half line, but you have like a constant quantity, meaning that all the mass which is supposed to go to the negative half line will be concentrated on a Dirac. And actually, that's the perturbation term of the energy. And that gives you the energy loss, okay? And now you can see how it evolves. For example, for the phase space, then you start initially like on a closed curve, then you stay a bit on the closed curve. And then when the size distribution is sufficiently spread out, then you go toward the steady state. And then the energy is decreasing first linearly, and then you have like an increased speed of decay of the energy. Okay? And so now it's just a zoom when actually the energy is decreasing. So you see the energy is decreasing when you have mass on C1. That's like the green color on the screen, okay? Okay, and now in phase 3, this is much more tricky because before we were using theorems based on the fact that epsilon was small and V and W were sufficiently big, and so we could neglect like some terms in the equation for the V and W, actually. But now, since in phase 3, the curve is really close to the steady state, which is of order epsilon, then we cannot use asymptotic perturbation to analyze the system. Still, what we have is a numerical result, and what we can see is that we still have oscillation, but it's much more like a harmonic oscillation, and it involves only the cluster of small sizes. The large sizes, so the distribution for the large sizes almost remain constant, and then it's only like an oscillation processes for the small sizes, okay? And then in phase 4, what we have is like the energy stays constant. Actually, the energy is not a Lyapunov functional, as I said, so it will not go to 0. It will stay constant. And then what we have is like the V and W are sufficiently close to their steady state, and you have only like the dynamics for the polymers, which is on a parabolic equation. Okay. So in conclusion, with this kind of system, we have a complete understanding about the oscillation for some specific case for the constant coefficients. We have like two kinds of oscillatory mode. The first one is one where we have like two time scales. So you have some kind of spike oscillation where almost all the time nothing happens. And then when something happens, it's on a really fast time scale. And the second one is when the V and W are close to their equilibrium values, and then you have harmonic oscillation. And okay, so one of the perspective is like to complexify this model in order to fit the dynamics of this model to the experiments. And this is what Uman showed you actually. So on the right, on the top right, you have the size distribution. You see you have a diffusion process. Then you have like some transport. And then you still see the transport process, but you still have a strong diffusion process. And what you see on the left is the phase space for V and W, where you have the convergence toward the steady state. And on the bottom, what you can see is like the second moment distribution of the second moment of the size distribution. And then you see like this plateau at the beginning is only due to diffusion. And when you have like this big decrease on the second momentum, that's due to the transport phenomenon. Okay, so what we want is to refine this kind of equation, complexify, add some terms in order to have a really good fitting with the experiments. The second perspective of this project is now that we have some new data on human size about patterns that are formed by the amyloid fibrils. And what we have is that now we are not in a closed environment, but we are in ex vivo environment where you have like slices of brain, and they can inoculate these slices. And what they see is like the black spots are the accumulation of prions. And now what we want to understand is actually the spatial dynamics of this kind of diseases. Thank you very much.